Y chromosome.
Yeah.
So let's turn it over to Emily and Brittany.
Alrighty. Hi everyone, I'm Emily Cutting.
I'm Brittany Weiss.
And we did a project where we looked at differentiating between the alleles of the gene Y150 20.6 on the Y chromosome in 4 different species of bears. Alright. When phylogenies are made, and they're based on, when phylogenies made on DNA are made, they're usually based on either nuclear or mitochondrial DNA. Mitochondrial DNA is often used because it's only inherited from the mother. And unlike nuclear DNA, it doesn't get all mixed up, so it's really easy to distinguish a single parental lineage, which is very helpful if you're trying to make a phylogenetic tree. But the problem with mitochondrial DNA is that it's only inherited from the mother. So it doesn't take into account male-mediated gene flow. And because of this, the National Fish and Wildlife Forensics Laboratory did a study where they looked at different microsatellites, different pieces of DNA with short repeating nucleotide sequences and compared them to see just how influential male-mediated gene flow was in the evolution of these bears. And they came across the problem when they tried to amplify the gene Y150 20.6. What happened is that instead of getting one product like they should, they got 2 products of differing length. This means that at some point during the evolution of these bears, this gene would have had to duplicate. And our goal was to make primers that would only amplify one of these alleles, so if they wanted to use this gene in the future, they could use it. And on top of that, we wanted to sequence our DNA and then compare these alleles to between the alleles in the same species and different species, see how similar or different they are, and hopefully make our own little phylogenetic tree. Oh, and these are the alleles, or not the alleles, the primers we use to amplify our gene. The National Fish and Wildlife Forensics Laboratory donated to us DNA samples from 4 species of bears. They gave us samples from the American black bear, the Asiatic black bear, the spectacled bear, and the sun bear. And we just want to give you an idea of where these guys are from. All right. The American black bear, as these guys have told you about, live in North America. The Asiatic black bear and the sun bear have some overlapping habitat in Asia, and our spectacled bear lives in South America.
So we actually have some sun bears at the Oregon Zoo up in Portland. I was recently there and I was able to get some first-hand experience watching these bears. We've got 2 bears up at the Oregon Zoo, and so these are the 2 bears that we have just up in Portland. These aren't the bears that we have DNA samples from, but I thought it'd be nice to have some first-hand photographs that weren't just stock photos. So here you see the 2 phylogenetic trees that we're going to be using for comparison. We've got the one based on nuclear DNA and one based on mitochondrial DNA. Both of them are looking at multiple genes, so they've got more than one gene, while our phylogenetic tree will only be using one gene. You can see that in both of them, the spectacled bear, the T. ornatus, is the outgroup. It's the least closely related to the other species that we're looking at. And then you can see that they also have the American black bear and the Asiatic black bear in both of these trees are more closely related than others. But other than that, the relationships aren't consistent between the 2 trees. And that's partially why the research into the Y chromosome gene was important, was to see how it compared to these 2 hypothesized phylogenetic trees. So we were able to get the DNA samples from the National Fish and Wildlife Forensics Laboratory, and what they gave us were already DNA extracts. They weren't blood samples or hair samples. And so our first step, once we received those DNA extracts, was to use PCR, polymerase chain reaction, to amplify our DNA samples. Polymerase is an enzyme that is naturally occurring in the body, and it's used to replicate DNA. And so using PCR in the lab mirrors what happens in the body naturally when it's making new DNA copies. So PCR is doing exponential duplication. For every time that you run the cycle, you're doubling the number of sequences that you have. And so we did 30 cycles of PCR. So that produces 1 billion times more copies than we started with. So we're going from a sample that already had many thousands of copies of the genome into over billions upon billions. And you're using a thermocycler. This is a machine that regulates the temperature of the environment that this DNA is in to control the polymerase enzyme and the DNA strands. So here you can see the PCR products that we got afterwards. We started with 7 samples, including 1 female. We did not expect the female to be amplified, as we are looking at a Y chromosome gene, and females don't have Y chromosomes. So we were expecting to get 6 amplifications. We did have 2 that we were unable to amplify, D and F. But we were able to amplify 1 sample from each of the 4 species that we were given, which we were really excited about.
So at this point, we just performed PCR, and in our tubes, we have a ton of both of the alleles. But we need to sequence these, and if we tried to sequence it now, we'd have a big gobbledygook because we're sequencing both alleles at once. So we had to separate these alleles, and to do that, we ligated these alleles into our PGM-TEZ vector, and then we transformed our competent E. coli cells by inserting this vector into the cells. And we took advantage of this vector, or 2 genes of this vector. This first one is that purple AMR gene. And what happens is that if it would give the bacteria the ability to be resistant to ampicillin. And then this other gene right over here, this orange one, is black Z gene, which would give the bacteria the ability to break down galactose. And what we did is we inserted our DNA right into the middle of that black Z gene. So what happens, it would make that gene non-functional. So we spread our plates with both ampicillin and galactose, and then we spread our E. coli we transformed onto it, and the genes, the bacteria that appear blue here were able to break down galactose. This means they took out the vector, but the vector did not have our allele in it. The white colonies here took up our vector that had our allele. And each one of these colonies has only 1 copy of our allele. So we took 3 samples, or we have 1 sample for 3 colonies on here, and hope that we got both of the alleles and not the same allele 3 times. After this, we took our samples, incubated them in broth overnight, and then we extracted our plasmid away from the rest of the bacterial cell and performed a sequencing reaction. The sequencing reaction is very similar to PCR, except instead of using just total normal nucleotides, a certain fraction of the nucleotides we used were called fluorescent dideoxynucleotides. And these are special because when you're performing the PCR, as soon as one of these dideoxynucleotides are incorporated into the DNA, replication stops. It can't go on, it stops. So after this has been repeated like hundreds and hundreds of times, you're going to end up with DNA that are different size, with these fluorescent deoxynucleotides. And then we put these into our API 310 sequencer, which separated them by size, and the sequencer was able to tell which deoxynucleotide stopped the replication of the allele, which gave us this little graph down here, which is our sequences.
All right, so here you can see the 8 different alleles that we were able to determine. We got 2 alleles from each of the 4 species that we were working with, the sun bear, the Asiatic black bear, the American black bear, and the spectacled bear. You can see that the 2 alleles for each species are very similar. They have a single deletion of the GA repeat, which is the STR region, the short tandem repeat region. And so the differences between the 2 alleles within a species are a deletion of 2 nucleotides, 1 G and 1 A, and that's the only difference between the 2 alleles for each species. If we go to the next slide, we compare the 8 alleles total between all the 2 alleles between each species, as well as the 4 total species. And you can see that before the STR region, before the GA repeats, the spectacled bear and the American black bear both have an insert of AGAC. All the species have different numbers of the GA repeats. And then at the end of the STR region, after all the GA repeats, the Asiatic black bear and the sun bear both have AT sequences. The spectacled bear has a single C nucleotide, and then the American black bear has an AT sequence.
All right, and here are phylogenetic trees again. This one in red over here is our phylogenetic tree. And the first thing you should notice when looking at these is the alleles from the same species are more similar to each other than any of the alleles from the other species. And another thing to look at is that the only similarity between all of these trees is that our spectacled bear, our T. ornatus, is the outgroup in all three of the trees, and apart from that, they're completely different. For example, like our sun bear and our Asiatic black bear in our tree are most closely related to each other, while that's not in any of the other trees. And this is probably because we only got a sample from one bear from each species, so it's just really hard to make a good phylogenetic tree using this small of data. I'm sorry about that. Oh, there we go.
All right, so in conclusion, we determined that we can't do a primer that will amplify only one of the alleles for these bears because the two alleles are so similar, and where the differences are is so far into the gene that the primer at the beginning of the gene won't pick up the differences. We also determined that the genes are probably too similar to use as an evolutionary or population marker, just because the differences between the species are so small that it might not be a good idea to be using those, and that in all likelihood a different Y chromosome gene is going to be a better subject for studies on population and gene flow. For follow-up research purposes, we did suggest that we could use the primer that's currently being used to probe the DNA and determine what the upstream sequence is, what the sequence of DNA just before the Y15020.6 gene is. And if that upstream sequence is different, then we could use that as a primer to continue studying this gene. And then, of course, identifying an alternate Y chromosome gene would be probably the most resource efficient for the National Fish and Wildlife Forensics Laboratory.
Okay.
So, do we have any questions?
Do we have any questions? Yes.
What was your favorite part about your research?
One of the things that was really exciting was when we took our sequences and ran them through a catalog of a bunch of different sequences that have already been published about. We did not find any matches. So our gene is very new research, and it hasn't been incorporated into a lot of the databases of the different alleles. So it was really cool to be one of the first people to be working with this gene.
So, this, was the difference between the alleles, was it that substitution, or was there an insertion that made the alleles different sizes?
Between the two alleles of the same species or between the different species?
Both.
Okay.
So between the two alleles within one species, it was just a single deletion of two nucleotides, or conversely, you can look at it as a single insertion of two nucleotides. And then when you're looking between the different species, it appears to be insertions or deletions, and they're not substitutions.
Do you think it would be useful to maybe like look at the size of the alleles rather than your sequences to?
They are very similar in length. You can see they go from about 125, just under 130 to just about 135. So they're so close in length that that's probably not useful.
I just want to add that I think your personal pictures of the bears just added very touch your presentation.
It was a coincidence that I was able to get them.
It
So,
works.
yes.
Nice job.
Anything else? Okay, thank you.
All right, well, thank you all again.